So welcome back, folks. In this part, we'll be talking about audition, or the ear. And it's really based on the understanding of the presence of different sound waves and what the structure of the ear is and how the ear picks up these waves and transmits that into an action potential. So sound energy is transmitted through gas, liquid, or solid. Okay, due to these vibrations, we are usually picking up sound through air. Okay, in parts where there is a vacuum, where there are no molecules whatsoever, we do not have the ability to hear. I'll go over the structure of the human ear really quick, just to kind of like a refresher here. So there are three parts. We have the outer ear, or the external ear, that is made out of the ear pinna or the auricle, and that's the funnel-shaped part. That's the part that we can see on the outside. And then it will funnel the waves into the external auditory canal, take the waves to the tympanic membrane or the eardrum. Once the waves pass the tympanic membrane, we are now inside of the middle ear. The middle ear is a cavity found within the temporal bone, and it's full of air. It should not be full of fluid. In the case, for example, of a middle ear infection or allergies, this part the cavities fills up with fluid, and that's why it's harder to hear if you have a middle ear infection or full of inflammatory fluid due to allergies, for instance. So again, the middle ear cavity is full of air with these three ossicles. They are named in order: malleus, incus, and stapes, or MIS, M-I-S. The stapes covers up a hole here that's known as the oval cavity, or the oval window. And that oval window will take these waves, so these ossicles are going to magnify the sound waves, and then take those sound waves through the oval window into the inner ear. The inner ear has these semicircular canals, a vestibule in the middle, and a cochlea that kind of looks like a snail at the end. The cochlea is the part that we hear with. These two portions are for different, for equilibrium. And then we have the eighth cranial nerve that is known as the vestibulocochlear nerve, made out of a vestibular branch carrying equilibrium and a cochlear branch carrying hearing. We also have the auditory, or the eustachian tube, and that connects the middle ear to the nasopharynx. Now, if we take, more of a diagrammatic or a schematic representation of the different parts of the ear. So again, this is your tympanic membrane. That is the middle ear with the three ossicles, the malleus, incus, and stapes. Here the stapes, you can see, would be vibrating against that oval window, taking those waves that have been magnified into the inner ear, and then those waves are going to exit the inner ear through the round window, and then they would go through that eustachian tube and leave the ear. And that's why we do not hear an echo, because those waves eventually exit out of the ear through the nasopharynx. Now for the different parts of the labyrinth, so this part right here, all of the semicircular canals and the vestibule and the cochlea, those are known as a labyrinth. The labyrinth has a bony part and a membranous part. So the membranous part, and that's the part in the middle, makes a duct known as the cochlear duct. And the cochlear duct is full of endolymph. That's a liquid found in there. While the osseous labyrinth, the part that is above the cochlear duct is known as the scala vestibuli, and the part underneath, or that tunnel underneath the cochlear duct, is known as the scala tympani. So if you follow, and that is both of these scalas, so the scala vestibuli and the tympani, are full of perilymph. Now if you take a look at how the sound waves would go, so they would be picked up by the external ear, by the auricle, pass through the external auditory canal, vibrate the tympanic membrane, and then these waves will be amplified through these three ossicles, pass through the oval window into the scala vestibuli, go all the way through, depending on the frequency and the vibrations. But let's say it went all the way to the helicotrema, which is the tip of the cochlea, and then passes through the scala tympani and exits out of the round window through the eustachian tube out to the nasopharynx. Here you can see the transmission of sound vibrations through the middle and the inner ear. So one more time, transmission, or the tympanic membrane will detect these waves, vibrate, and then those vibrations will be amplified through the ossicles. Now you can see the waves go through the oval window and they enter the scala vestibuli, depending again on the frequency waves. So high frequency waves will pass through the cochlear duct and then out through the round window right away, while the lower frequency vibrations will go all the way to, will go further down that cochlea, more towards the tip or the helicotrema, and again down in the scala tympani out through the round window. So at this point, we have been talking about waves and different chambers and so on. We have not really reached receptors. So this is where we will find the receptors. Remember that cochlear duct. This is where the receptors for hearing are. So we are looking at a section of the cochlea where this right here is the cochlear duct. Above it was your scala vestibuli. Underneath it was the scala tympani. Now the cochlear duct has the receptor, and the receptor is known as the organ of Corti. Corti is the person that discovered it. So here you can see a bigger image of the organ of Corti, which basically is this cell, these cells with little hairs. Hairs have cilia, and those are known as stereocilia. Those little hairs are going to touch a membrane known as a tectorial membrane. And as the waves pass through the scala vestibuli and the tympani, that is going to lead to the bending of these hair cells. And as these hair cells bend, they are going to either depolarize and so on, and that would lead to their stimulation. So here you can see how we transmit, or how we change these waves, sound waves, into an action potential. So again, when you have these stereocilia that are bending back and forth because of the passage of those sound waves, that is going to open up potassium channels. So you'll see here that depolarization in the case of hearing is a little bit different. Everywhere else, we open up sodium channels first. But in the case of hearing, we open up potassium ion channels. And because of that opening, that's going to lead to the depolarization. So you see here, this is opposite of what happens in all of the other cells in the body. The neurotransmitter between that receptor and between the neurons is glutamate. You definitely do not need to know these numbers, but I just thought it's very interesting to kind of figure out what are the different levels of hearing. So we can go from just audible breathing sounds. You can see, for example, vacuum cleaner or a hair dryer is a little bit louder, all the way down to like in a rock concert where these are the levels of decibels that you can hear. Know that the threshold of pain begins at 125, so sometimes these rock concerts can get pretty crazy. A jet takeoff, when you're only at 75 feet distance, that can lead to, that can actually lead to the rupture of the tympanic membrane and could lead to permanent damage. So again, this is just here for kind of, personal interests. Now, what is the neural pathway of hearing? So the cochlear nerve fibers, remember we've got the vestibulo-cochlear nerve made out of two different parts, the vestibular part for equilibrium and the cochlear part for hearing. So this is what we're talking about here. We have not talked about equilibrium yet. So the cochlear nerve will have to send the impulses up through the brainstem, and then from the brainstem it would have to go to the thalamus, and then from the thalamus, it would go to the auditory cortex and the temporal lobe. So just like any other sense, we've got these three different neurons. We have the primary neuron, and that's the found in the cochlear nerve. We have the secondary neuron where we're transmitting to the thalamus, and then our third neuron from the thalamus to your temporal cortex. Now let's go a little bit deeper in order to figure out, talk about equilibrium. So this labyrinth has, as we mentioned, has the osseous labyrinth, and that's the bony part on the outside. And then on the inside, we have a membranous labyrinth. Okay, so it's kind of coated on the inside with a membrane. Remember that the membranous labyrinth is full of endolymph, while the osseous labyrinth is full of perilymph. Now we'll break down, so we talked about the cochlea already, so we can kind of put that to the side for a minute. We are going to talk about the semicircular canals and the vestibule. So these semicircular canals have little, kind of like little pouches at the end. These pouches are known as the ampulla of the semicircular canals. Inside of these ampullas, we have these little triangles known as the cupulas. Okay, this is where the receptors are. The semicircular canals detect changes in angular acceleration. So things, for example, like going on a roller coaster or twirling like in ballerina dancers or figure skating and so on. So these changes in movement are detected through these semicircular canals. The vestibule also has that membranous portion, and it's made out of a utricle. If I can get my cursor to work, there we go. So the utricle on the up here, and this part is the saccule. These also have receptors that we'll talk about in a bit, and they detect linear acceleration. So for example, walking or being in a car. Okay, so where you're going pretty much in a straight line. So let's take a look at the cupula and the ampulla. And if you don't remember what those are, this is what we're talking about. So we're talking about these ampullas of the semicircular canals and the cupulas in them. And what do these receptors look like? They look very much just like the receptors for hearing. Again, we have hair cells with stereocilia. And as we move around, we go on that roller coaster ride, these hair cells are going to bend back and forth. So you can see the bending back and forth, and that is what stimulates them and will change that acceleration into a graded potential. Okay, and that's what is going to activate in this part, in this particular part, we are going to send the impulses on the vestibular nerve. Now for the utricle, and again, if you do not remember what the utricle is, that is the part that is found within the vestibule, which is the middle portion of this labyrinth. And that again is responsible for your linear acceleration. You can see here the exact same concept where you have hair cells and stereocilia that are embedded within this gelatinous material. Again, as you see this guy bending down, the gelatinous material pulls down with it due to gravity, and these hair cells bend, hence leading to the formation of a graded potential. So this is how you activate these receptors. So what is the pathway for vestibular information? Well, first off, we'll talk about why we need vestibular information. It's important to control eye movement. You want to be able to control your posture and balance. So why do I need all this information? Well, you need to figure out, are you moving forward? Are you doing a cartwheel? Where are you in relationship to everything else? Okay, so you want to be aware of your body position. Are you going fast or slow? So aware of acceleration and spatial information. All of that is going to help us maintain our upright posture and balance. It also helps us in controlling eye movement. So if you lose these receptors, if you lose the function of the vestibule, that could lead to nystagmus, and nystagmus is this very fast eye movement that is not controlled. Vestibular nerve fibers will then go, it's getting very redundant at this point, where we'd have to go through the brainstem and then to the thalamus. From the thalamus, it will go to the vestibular centers that are found in the parietal lobe. Vestibular information is obviously integrated with other sensory information coming from proprioception, and if there's a mismatch, that would lead to motion sickness. So let me explain how this works. Let's say, for example, you're in a car and you're looking out of the window. Your brain is getting impulses from your vestibule that you are moving, okay, so linear acceleration, and it's also getting impulses from your eyes. So you're seeing, things going by. So the impulses coming from the eyes, the messages are that you're moving. So these match and you're fine. Now, you've decided that you're going to start reading a book while you're in the car. So your vestibule is still giving impulses to your parietal cortex that you're moving. But your eyes are looking at a fixed book. So your eyes are sending impulses that you are not moving. Now there's a mismatch, and that could lead to motion sickness. So the motion sickness happens due to the mismatch between the information coming from the vestibule and the information coming from other senses. So that covers the auditory part, and I will see you in the next portion.